Hey guys, and welcome to our lab practical review, our number 1. So in this review, I'm going to go through the key components of each protocol that we've done so far. So your lab practical is going to cover experiments 1 through 6. So I'm going to point out some key components, and then also just remember that this video is not all inclusive. It's more or less an idea on to help you gather your thoughts and gauge what do you feel is relevant for studying. So before we even start with microscopes, I want to go over lab safety real quick. So with lab safety, I want you to know what personal protective equipment is. So this is stuff that you wear in order to protect yourself during lab. So what you wear during lab in order to protect yourself is gloves, lab coat, and goggles. And then also, if you have long hair, you pull your hair back, but that's not personal equipment. So just be aware of that is what lab safety is. So I just want you to remember that. And then also, if I were to give you an image, I would want you to be able to tell me what is wrong with this image in terms of lab safety. So whether or not if the person is not wearing their goggles, is not wearing their lab coat, or their hand is too close to the flame, like whatever it may be, or no, not wearing gloves. So just be able to point that out. That will be test questions. Okay, so I believe that was all I wanted to cover for in terms of lab safety. So with that, let's go ahead and get started with experiment 1, which was microscopy. So with microscopes, I definitely just want you to know the general big picture idea of, of a microscope. So like this is a microscope, and then also be able to calculate total magnification. So total magnification is always the objective lens times the eyepiece lens. So the objective lens is either going to be 4, 10, 40, or 100. And then the eyepiece lens is always going to be 10. So if I were to ask you, be like, calculate the total magnification of a specimen on the blue objective. So you would need to know that the blue objective is, so if I were to say calculate, so on the blue objective, which is, you need to be able to tell me that that is the 40x objective. So with that, using the equation above, what we'll do is, sorry guys, I cannot type today. My goodness. Okay, so if we wanted to calculate the total magnification, it would be, according to our equation above, it would be 40 times the eyepiece lens, which I said was always 10. And then that will equal, so 40 times 10 is 400x. So that is how you calculate total magnification. So I will expect you to do that. And then also another thing I want to point out is resolving power. I want you to know the concept of what it is. So that's definitely probably, in my opinion, the most important concept of microscopes. But definitely like know what depth of field is and all that good stuff. But to me, the most important one is resolving power. And I believe that was it for the microscope lab that I wanted to point out. And then actually one other thing, I do want you to know the parts of the microscope. I do want you to know the function of each one. So the big functions of each one is the fine focus, the coarse focus, and the iris diaphragm. So I definitely want you to know, if you're going to know any of them, I want you to know at least those 3. So what is the purpose of them? Okay, so definitely know like why do we use them. So I believe that will wrap it up for microscopes. So with that, let's go ahead and transition into experiment 2. So with experiment 2, you need to know the concept of quadrant streaking. So what is the purpose of quadrant streaking? And with that, that will give you the concept of pure colony. So what defines a pure colony? What makes a pure colony a pure colony? And then also, whenever we are transferring microbes from one media to another. So what is that called? So that is called subculturing. So you need to know the concept of subculturing. And then also, the skill that you need to show me on the exam is how are you going to streak a plate. So you're going to simulate doing a quadrant streak, so definitely know your technique, watch videos on it. If you need to, you can always ask me for like an extra plate to practice on. That's completely fine. But definitely know the concept of quadrant streaking and be confident in doing it. So whenever I say that I'm going to expect you to streak a plate, I'm going to have you literally show me streaking a plate, showing all the steps, meaning flaming the loop, getting broth, getting your culture, and then flaming in between. So everything that involves streaking, you need to be able to do on the day of the exam. Okay. And then also, from subculturing, we also looked at different forms of how do certain things grow in certain media types. So I definitely will want you to be able to identify these guys. So if I were to ask you like if I were to label these test tubes like 1 through 6, I would be like test tube 6. Like which of the following slants shows rhizoid growth? So I would want you to select test tube number 6. Okay, and same idea with broth growth. So I would definitely be able to want you to identify that. And then as well as the form type in terms of plate. And then elevation, I'm not too concerned about elevation because that's very, very hard to get on an exam. So elevation, you don't really need to worry about for the test, but definitely be able to identify form type as well as margin type, okay? So be able to identify those. So if I were to like give you a picture of a plate, and I would be like, I would box or indicate like this part of the plate, I would want you to be able to identify the form type and margin type based on the photo that I gave you. So definitely practice that. I won't give you anything too extravagant. I will give you probably something very close to this, so it will be very, very easy to identify. So I think that was the big picture for subculturing. Let me just make sure. Yes, so that was the big picture of subculturing. So like I said before, with subculturing, you need to know the context of what is subculturing, what is the whole purpose of it, and then from quadrant streaking, why do we do that? And we do that so we can get a pure colony. And then the whole purpose of getting a pure colony is because we want to make sure that we are getting a single species of cell, essentially. Because theoretically speaking, if you have a colony all by himself, that colony arose from one single cell. It did not arise from multiples. So that's the theory on why we want a pure colony and what we do to get it, which is quadrant streaking. And then don't forget, you will have to show me your quadrant streaking skills. So be practicing at home. Literally just practice with, you can even make it with Jello and then use a pencil. And then just practice being like flame, flame, flame and stuff like that. So, but don't stress too much about that, guys. That's not even, I think it's only like 7 points on your exam, if that. So don't stress about it, okay? And then also be able to identify different form types in terms of slants, broth, and then as well as plate form. So this is on for the plate right here, and then be able to identify margin type. Okay, so that was the big picture for subculturing. So let's go ahead and transition into experiment 3, which is where we were simple staining. So simple staining, this is where we did, simple staining is where you only use one reagent. So definitely be able to tell me the definition of simple staining, and then the differences between a basic stain and an acid stain, and examples of each of those types. And then also, what do you do whenever you are preparing a bacterial smear? So there are three main steps that you do whenever obtaining a bacterial smear, and that is, what you do is you smear it, you air dry it, you heat fix it, you stain it. So, but in preparation of just the smear, it only includes the bacterial smear, the air drying, and the heat fix. Okay? So, definitely know that those are the three things that you need to do prior to actually applying the stain. Okay? So, with that, that will bring us to the idea of all the different stains that we use. So definitely know what stains you use for which type. So if you are gram staining, you're going to use like Graham's iodine, you're going to use crystal violet, you're going to use saffron for counter stain. So definitely know that. And then also from, I believe I have it on here. No, I do not, but I have it on the review. So definitely these tables, guys. So I provided these tables because I think they give a good overview of each stain type. So like with gram staining, be able to tell me the primary stain is crystal violet, and then the counter stain is saffron. And then based on if you have a gram positive or a gram negative, be able to tell me what will be the end result in color of the bacteria. And that also goes for in terms of acid fast staining and spore staining. So definitely be able to understand these tables and also the actual reagents that you use for each one. Okay? Okie dokie. And then also from experiment 3, I will definitely want you to know the difference between cocci and bacilli. I'll definitely want you to know that, okay? And then throughout the exam, I will have a projection going that will have a picture of a stain, and from that I would want you to identify the morphology. So the morphology questions, so like the cell shape and cell arrangement, I will have a PowerPoint on cycle, and then it will show just various stainings, and from that I would want you to tell me, based on that stain, what is it? Like, what's the morphology? Like, is it, like, are they bacilli? And if they are bacilli, are they diplobacilli? Are they strep? Are they, that's pretty much the only two that I'll ask you. I won't ask you any of these. And then also like with cocci, like, are they diplococci? Are they strepcocci? Are they staphylococcus? So definitely be able to identify different morphology types. I will not ask any morphologies outside of the ones that we have seen in class. So for example, we have not seen spirillum in class, so I will not ask that. So I will only ask the ones that we've actually seen in class. And the ones that we've seen in class is single cocci, diplo, strep. We might have seen tetra, but not really. And then Staphylococcus, and then with bacilli, we've seen bacilli, diplobacilli, and strep bacillus. So those are the only morphologies I'll ask you, so be able to recognize that from stains. So literally just like Google would be like, gram stains of diplococcus or something, just so you can familiarize yourself with them. And then also, like, I may ask you, staining wise, what does a purple cell mean and what does a pink cell mean in terms of gram staining. So if you have a purple cell and it's gram stained, that means you have a gram positive. If you have a pink one, that means you have a gram negative, okay? So that is primarily for simple staining. Now for differential staining, it kind of goes with the same idea. So, but with differential staining, I definitely want you to know, where is it? This table. So, I'm sorry, not that table. this one. So the acid fast and the spore one. So I know we weren't able to see the spore, but I did send out results of what should have happened. So definitely look over that. If you have questions, please ask me in lab. Please don't ask me the day of the test. So definitely ask me about it. But the overall idea is that you will see something. So let me go ahead and actually just pull up the key for it so I can show you a little bit better. We'll do endospore staining. So with the endospore staining, I sent out this table. So the idea is that the red and the green, so they're kind of bluish in my opinion, but the blue-green cells, these are the spore cells that you'll see. And then the red ones are the vegetative, meaning that those are like the active cells that we do see. So this is what you would have seen if we did have those B series stress plates. So definitely be able to recognize that, okay? So know the difference between what does it mean to be a blue-green cell as compared to a red cell in the context of endospore staining. Okay? Okie dokie. So like I said, I believe that's the big picture for the staining types. Also, be able to tell me what is a potential, like for acid fast staining particularly, what organism in general is acid fast staining normally always used for? And the answer is tuberculosis. So, or microbacterium actually is a better answer, but tuberculosis is a species of microbacterium. But definitely be able to tell me at least one, maybe two organisms that can be identified using each of the staining types. So like with gram that's a little bit more broad. Just for the most part, be able to tell me that with gram staining you can identify between a gram positive and gram negative cell. And then with acid fast staining you specifically can determine a microbacterium. And then with endospore staining you can specifically see if spores are present. So I just want you to know what's the big picture of each one. The acid fast staining specifically is probably the only one that can give you like a true narrowed in of the genus, and that's specifically microbacterium. Okay guys? Okie dokie. And then also be able to tell me, what do they all have in common? What step do you have to do in every single one? So every single time you stain, you are always heat fixing of some sort. Okay? Whether if you're doing it at the very beginning or at the very end, all of these stains, specifically with differential staining, you're always heat fixing of some sort. Okay? Okie dokie. I think that will wrap it up for experiment 4. So let's go ahead go to experiment 5. So this was our big mama experiment. So this is where we identified different growth requirements and how to actually classify them based on how do they grow. So definitely be able to differentiate what is a psychrophil, a mesophyll, a psychotroph. So be able to tell me what is a psychotroph, and then what's a thermophil, and then a hypothermophil. I take that back. Don't even tell me about psychotroph. I don't care about that one. So just know the phils. So know psychrophil, mesophyll, thermophil, and hyperphil. So definitely be able to differentiate between those. Know the temperature ranges of each of those guys. Okay, and then also, where is it? With pH. So specifically with the pH, I want you, same thing with the temperature, I want you to be able to tell me what makes it either an eosinophil or an alkalinophil or a neutrophil. So I want you to tell me that. What are the pH values for each of those? So that's the big thing in terms of pH and temperature. What else? Let me see. Okie dokie. And then also, one of the things that we did, some of y'all may have done it, some of y'all may not have, but that's okay. So we used for this part of the lab with the pH and the growth salt requirements, what we used was a spectrometer. So what I want you to know is what was the purpose of the spectrometer. So we kind of talked about it a little bit in lecture. So spectrometer is an indirect form of measuring your culture. So it measures it through the use of light, so turbidity. So what it does is that the spectrometer will shine a light through your test tube, and then based on how much light goes through, we'll determine how turbid it was. So the more turbid, meaning the less light that went through, means that your culture grew a lot more, as compared to the other way, where if you had less turbidity, meaning that you had more light that went through, meaning that you had less growth. Okay? So definitely know the concept of a spectrometer. I'm not going to expect you to use one on the day of the test, but I do want you to know the purpose of it. Okie dokie. So with that will bring us to the next one. So with these guys, I'm definitely going to expect you to identify classes of oxygen requirements. So like with the first one is an obligate anaerobe, meaning that it requires oxygen to grow. And then obligate anaerobe, meaning that it does not want free oxygen. Number 3 is a facilitated, meaning that they can grow either direction. And then, oopsies, and then a number 4, this is called a microaerophil, meaning that it only needs a certain amount of oxygen. It only needs a little bit, as compared to aerobes, where they need all the oxygen they can get, whereas number 4, they only need some. And then number 5, an aerotolerant anaerobe, it doesn't require oxygen, but it won't die if oxygen is not present. So technically it can grow anywhere within the tube. So definitely be able to identify that different types of classification with oxygen. So be able to tell me those 5 types. And then also I think that's actually it. Just kidding. So just in the context of that, I won't ask you anything about the gas pack. So I won't ask you anything about that. Okay, and then one other thing that it kind of ties into experiment 5, but it's a little bit, it's kind of separate, but it's kind of like a case study. So I'll give you a scenario, and then based on the scenario, I would want you to name some conditions that you would give this this organism in order to promote growth. So based on the information I gave you, I would want you to tell me, what would you do to promote growth, whether it be change temperature, change salt content, take away oxygen, or give it a lot of oxygen. So just tell me, what are some conditions that you would do that could help facilitate growth of this organism based on certain observations that I give you. Okay, so that is for experiment 5. Experiment 6 is over gram-positive, so a lot of this stuff we've actually kind of already done. I don't actually believe I asked you anything directly about it. let me just double check. Let me look over the exam real quick. It doesn't look like it. So I don't ask you any direct questions about gram-positives, but I definitely want you to know the concept of what is a gram-positive. So like gram-positive, like what was an example of a gram-positive? So that's E. coli. So know that he'll stain pink. Yeah, that's pretty much it. So actually, you don't even need to really look over this one for experiment 6, because I believe we have covered probably about 90-95% of the exam, guys. So definitely look over it, look over the review. So let me actually pull up the study guide just so I can double check for my own sake that I did cover everything for y'all. Okay, so I think I did. But if you have any questions, definitely please don't hesitate to ask. In my opinion, the lab exam should be a lot more easier and a little bit more direct, especially since we've been physically doing it in lab. But if you have any questions, definitely please ask me, guys. And I believe that's it. So I hope this helped y'all, and I will see you in class.